and welcome to the video abstract for our paper titled A Nanostructural Investigation of Glassy Gelatin Oligomers. This work was carried out in collaboration between the universities of Bristol, Santiago and Nottingham, as well as food concept and physical design. The Positron work was carried out at the NewJournalofPhysics laboratory, which you can see just behind me. Our paper explores the effects of water and glycerol on the molecular packing and thermodynamic state of low water content gelatin matrices by combining positron annihilation lifetime spectroscopy with a number of complementary techniques.
Gelatin is one of the most versatile and widely used biopolymers with a vast array of applications across the pharmaceutical and food industries. Its thermoreversible gelation, as well as its ability to produce strong films with adjustable morphologies, make gelatin an invaluable raw material for the manufacture of hard and soft capsules. These capsules are used to encapsulate active compounds in order to protect them from external influences and achieve their targeted release. One of the main issues in the design of encapsulating matrices with optimal barrier properties is the molecular mobility through the matrix, which in the glassy state is given by the density of the molecular packing. The irregular arrangement of molecules in the glassy state results in a certain amount of local free volume, which consists of a large number of sub-nanometer sized elements or holes. The size of these holes can be measured directly by using positron annihilation lifetime spectroscopy. By understanding the effects of low molecular weight diluents on the molecular packing and thermodynamic state of gelatin matrices, we can eventually produce encapsulants with optimal barrier properties. In positron annihilation experiments, a positron is injected into a material and we measure the length of time until it annihilates with one of the material's electrons producing gamma rays. Sodium-22 is used as a source of positrons since during its decay, a prompt gamma ray is emitted simultaneously with the positron. In molecular materials, a substantial fraction of the injected positrons form positronium. This is a metastable positron-electron bound state which can localize in a free volume hole, where it remains throughout its lifetime. The lifetime of the longer-lived orthopositronium is environment dependent, and it delivers information pertaining to the size of the hole in which it resides. The size of the free volume hole may be calculated by using the semi-empirical Tau-Eldrup equation. Positron annihilation lifetime spectroscopy measurements are made using a conventional fast coincidence system. This consists of fast-timing electronics connected to two identical photon detector units. One to detect the photon associated with the creation of the positron, and the other to detect one of the gamma photons emitted during its annihilation. For the Pounds experiments, gelatin films are stacked to produce a thickness of approximately 1 mm, which is enough to stop 99% of the positrons. Two identical gelatin film stacks are then placed on either side of the source, and the sample source assembly is sealed in an airtight sample holder. With a current experimental setup, we can take measurements in the temperature range of 100 to 430 Kelvin. Now, let's briefly summarize the main findings of our paper. It is clear that glycerol and water have very different effects on the molecular packing of the gelatin matrices. Both in the glassy and rubbery states, we observe complex changes in the average hole size as a function of the level of hydration, whereas over the concentration range studied, glycerol acts as a packing enhancer, causing a systematic reduction in the average hole size. Both molecules significantly reduce the glass transition temperature of the matrices, and this plasticizing effect may be modeled semi-empirically by using the Gordon-Taylor equation. In the glassy state, we observe a rapid increase in the hole size of the gelatin matrices as a function of increasing water content. The water molecules plasticize the gelatin matrices by binding to the polar side groups of the polypeptide chains. At higher water content, water molecules form clusters, and we measure a decrease in the average hole size of the gelatin matrices. For matrices with well-defined water contents, we observe a nonlinear decrease in the average hole size as a function of increasing glycerol content, which indicates a non-ideal packing behavior in the glassy state.